Hi everyone! Okay, so in this video we are going to be talking about protein digestion and absorption. So as you already know from your GCSE studies, proteins are digested first in the stomach and then in the small intestines. So we're going to start off by focusing on the digestion part, and we know that proteins are long chains of amino acids, and we know that there are 20 amino acids in nature. And if we just take a minute to look at the basic structure of a single amino acid, just to remind ourselves. And we know that amino acids are joined together by peptide bonds. The joining together of amino acids by peptide bonds is called a condensation reaction and forms water. So, what this shows us then is if you have any chain of amino acids, no matter how long, at one end you will have the amine group, and at the other end you will have the carboxyl group. So any chain of amino acids has an amine end and a carboxyl end. So when we draw a long chain of amino acids, we might choose to draw them just as different shapes, just to represent the different types of amino acids that are present in that particular peptide chain, and each of the lines represents a peptide bond. So this is our chain of amino acids, and at this end we would have the amine group poking out, and at this end we'd have the carboxyl group pointing out. So every amino acid chain has an amine end and a carboxyl end. That will be important later. So protein digestion begins in the stomach, and it begins in the stomach with an enzyme called pepsin. Pepsin is an endopeptidase. Let's just look at that word for a minute. Endo meaning inside, and peptidase meaning it breaks peptide bonds. So what is an inside peptide bond? Well, it's basically just any of the ones here, any of the ones that are not on the outside. So these bonds here, these are the terminal peptide bonds, so these will be broken by exopeptidases. We'll talk about those later. But endopeptidases break bonds in the middle, so we're talking about these ones here in the middle to create shorter peptide chains. So there's loads and loads of new language that we're going to be introducing here, and all of them sound quite similar. Basically, we're going to be using this term peptide a lot. So if we think of any chain of amino acids, we call that a polypeptide, and these are broken down by endopeptidases into shorter peptide chains. Now the term for shorter peptide chains is oligopeptides, and you also know that if they're broken down into peptide chains that contain two amino acids, these are called dipeptides, and we'll be getting onto those later. So in the stomach, pepsin, which is an endopeptidase, breaks down large polypeptide chains into smaller polypeptide chains, or oligopeptides, by breaking internal peptide bonds in the chain. Now I'm going to go on a very slight aside here. This is not necessary for A-level, so feel free to ignore what I say for the next couple of minutes. But pepsin is an enzyme that's produced by cells in your stomach, which are called gastric chief cells. Now, pepsin is a protease, and as you know, many of the things in your cells are made of proteins, so it's actually quite dangerous for a cell to contain internally a protein digesting enzyme. So what they actually do is they produce an inactive precursor called pepsinogen. Pepsinogen is essentially pepsin, but where the active site is covered up. So when pepsinogen is released into the stomach, hydrochloric acid interacts with pepsinogen and converts it into pepsin. So pepsinogen needs to be activated to become pepsin. And then once it's in the stomach, it's the hydrochloric acid that does that. And then the stomach lining is surrounded by a layer of mucus, and that thick mucus protects the actual epithelial cells of the stomach, or the lining cells of the stomach, from the activity of the pepsin enzyme. So hydrochloric acid in our stomach performs three major roles. One is to kill any pathogens that might be present in the food that we eat by creating this acidic, hostile environment. Two is to activate pepsinogen into pepsin. And three is to provide the correct pH for the activity of pepsin. So the optimum pH for the activity of the pepsin enzyme is pH 2, which is the pH of the stomach. So why is pH 2 the correct pH, or the optimum pH for pepsin to act? Because that's the pH of the stomach. So it's kind of like a chicken and egg situation, right? But the stomach needs to be a low pH to protect us from infection. And over time, the pepsin has evolved, adapted to be able to function at its optimum in that pH. So that's not actually necessary for the A level, but I think it's still interesting to know. So in the stomach, pepsin is hydrolyzing these large polypeptide chains down into shorter polypeptide chains. Then, after a few hours, food can be in the stomach being churned for about 8 hours, and then it goes through the pyloric sphincter, which is the circular muscle at the base of the stomach, into the small intestine. The small intestine is slightly alkaline. In these conditions, the pepsin is denatured. So pepsin is no longer functioning. And so the second stage of protein digestion occurs here. And now we have the endopeptidase trypsin. So trypsin is actually released in an inactive precursor form called trypsinogen, for similar reasons that we talked about pepsin and pepsinogen earlier. But it's released from the pancreas. So the pancreas releases into the small intestine via the pancreatic duct digestive juices. Now, these pancreatic juices consist of alkaline fluid, which mainly contains bicarbonate, and enzymes. So it consists of a mix of lipases, proteases, and carbohydrases, but we're going to just focus on the proteases right now. So it releases trypsin. It also releases exopeptidases, and we'll also talk about exopeptidases in just a moment. So trypsin continues the functions of pepsin. So it's hydrolyzing these polypeptide chains into smaller and smaller peptide chains. What is the purpose of this? This is to create more endpoints. So we're essentially increasing the surface area for the action of enzymes, because if you've got a polypeptide chain and you're making it into smaller and smaller polypeptide chains, like for instance, as we can see here, you've now got more endpoints which exopeptidases can act on. And let's just take a moment to talk about exopeptidases. Exo meaning outside. So exopeptidases break the terminal bonds in any peptide chain. What do we mean by the terminal bonds? We mean the ones on the end. So in this case, say we had a long polypeptide chain like this one. We're talking about these ones here. These are the terminal bonds. So remember how we said before that on one end you've got the amine group pointing out, and on one end you've got the carboxyl group pointing out. So exopeptidases that break the terminal bond at the amine end of a peptide chain are called aminopeptidases. So we have two types of exopeptidases. We have aminopeptidases and carboxypeptidases, and these release individual amino acids that can then be absorbed. And we'll talk about absorption a little bit later on. However, you will often end up with chains of amino acids that are 2 amino acids long. Dipeptides. So in order to break down a dipeptide, you need a dipeptidase. Dipeptidases are found attached to the membranes of the cells lining the intestine. These are called membrane-bound dipeptidases. So if we just imagine that this is a cell of the small intestine, this is the lumen of the gut, so all of our partially digested food is moving along here, and we've got our blood capillaries down here. So this is our basal membrane. This is our epithelial cell, and our epithelial cell has infoldings on the lumen side membrane. These infoldings increase the surface area for both absorption and for the attachment of enzymes, and these infoldings are called microvilli. So if we just put this into context of where we are, we know that if we think longitudinally along the lining of the intestines, we know that the intestines are lined with villi, which are infoldings of the surface of the intestine, and these villi have lots of blood capillaries. Running all through them. And we also know that villi have lacteals, which are open-ended ducts that absorb tissue fluid and also absorb fats. You have to forgive my somewhat terrible drawing. And where are we? Where are these epithelial cells lined with microvilli? They are here. All along the surface. That's where we are when we're talking about this cell. Okay. And then what's further down out here? Well, down here we've got muscle. We've got smooth muscle. We've got longitudinal and circular smooth muscle that enables contraction of the small intestines to move food along. This contraction of the small intestine is called peristalsis. And then we've got the serosa, so we've got connective tissue. This connective tissue just joins the small intestine to the rest of your abdomen tissues. So we've got all these layers. These layers also contain nervous tissue and all sorts of different types of tissue. So that's kind of just a little bit of context so you understand where this cell is. Okay, so attached to the membrane, the cell surface membrane, you have got membrane-bound dipeptidases. Membrane-bound dipeptidases break the peptide bond between a dipeptide to produce individual amino acids. Now, the benefit of them being here is that it reduces the distance for these amino acids to then be absorbed into the epithelial cell. So we've talked about a number of different enzymes involved in protein digestion. We started off with the endopeptidase pepsin in the stomach. Then the endopeptidase trypsin in the small intestine. Then the exopeptidases, carboxypeptidase and aminopeptidase, and then dipeptidases. And I realize that at this point I sound like I've gone insane and that I am making up a number of these words, but I promise you I am not. Why do all these enzymes exist? They just essentially exist to cut the larger chain down into smaller and smaller and smaller chains until they are single amino acids, and those amino acids are small and soluble, and now they can be absorbed. We can take them into our bloodstream like little Lego pieces, and then we can build them back up into big polypeptides again, into the things that we want them to be, like muscles and enzymes and hormones and skin and keratin and a million other things in our body. But we need to make them small enough so they can get across our intestinal lining, so they can get into our bloodstream, and then they can travel through our bloodstream to all the places in our cells, and then the cells in our body can use them for stuff. So this is like breaking stuff down just so you can build it back up again later. So the next thing that we are going to talk about is protein absorption. So how do we then get these amino acids from the lumen of our gut into our bloodstream? So we need to travel from here to here. So this is a two-step process. So let's first start by talking about the active transport. So what essentially happens is, up here in the lumen, we've got lots and lots of amino acids, and we've got lots and lots of sodium ions. In order to draw the amino acids into the epithelial cell and then into the bloodstream, we need to use a co-transporter. So present on this membrane here, if we just were to focus here on this membrane for a moment, we would see that there is a co-transporter molecule embedded in the membrane. This is a transport protein. It's a co-transporter. That means that it has two binding sites. The first binding site is for sodium ions, and the second binding site is for a particular amino acid. This process of moving the amino acid into the epithelial cell requires also a sodium ion to bind. This is facilitated diffusion. That means that it's moving down a concentration gradient. The concentration gradient is of the sodium ion. So the sodium ion is moving down its concentration gradient. And as it moves, it's bringing the amino acid with it. So the amino acid is being drawn alongside the sodium ion as the sodium ion moves down its concentration gradient. So the absorption of amino acids is reliant on generating a sodium ion concentration gradient. So when these two binding sites are filled, this causes a change in shape of the transport protein, and both the sodium ion and the amino acids are moved into the cell. But this only works if we have a concentration gradient for the sodium ions. So in order to maintain this concentration gradient down here on the basal membrane, we have a pump which pumps sodium ions out of the epithelial cell and into the tissue fluid, where they will travel into the capillary and be moved away. So let's take a look at that. This is a co-transporter. It moves sodium ions out of the epithelial cell, and it moves potassium ions into the epithelial cell via active transport. So just think about that for a minute. This movement of the sodium ions into the capillary requires ATP. And that ATP comes from respiration, aerobic respiration. So if there was no aerobic respiration, this cell would run out of ATP, and then this sodium-potassium pump would stop working. There would no longer be a concentration gradient for the sodium ions between the lumen and the epithelial cell, and this would stop happening. The amino acids would stop being co-transported into the epithelial cell because there is no diffusion gradient for the sodium ions to move into the cell with the amino acid. So let's just check that we understand this concentration gradient. So we have a higher concentration of sodium ions in the lumen in comparison to in the epithelial cell. So the sodium ions move down their concentration gradient via facilitated diffusion. And as they move down their concentration gradient via facilitated diffusion, they bring the amino acids with them. This concentration gradient is maintained by the sodium-potassium pump, the active transport of sodium ions out of the epithelial cell and into the bloodstream, where they are then taken away. So once the amino acid is in the cell, it then needs to travel from the epithelial cell into the bloodstream itself. So now we need to look at the basal membrane again. Also embedded in the basal membrane are carrier proteins that will transport the amino acid out of the basal membrane into the tissue fluid, where they can then travel into the bloodstream and be taken away. And that's it. So that's how amino acids are absorbed into the bloodstream. There are carrier proteins embedded in the membrane on the lumen side for co-transport via facilitated diffusion, and then there is the sodium-potassium pump on the basal side, and then there are carrier proteins for facilitated diffusion of the amino acids. The amino acids obviously just diffuse via simple diffusion through the cytoplasm of the epithelial cell. So just a couple of things if we're thinking about how to answer exam questions. We've already talked about what would happen if we didn't have enough ATP. So you could get questions about respiratory inhibitors, for instance, or a lack of glucose available to the cell and therefore it can no longer do aerobic respiration. This would always affect the sodium-potassium pump, and that would always affect the generation of the concentration gradient of the sodium ions and therefore prevent the facilitated diffusion of the amino acids from the lumen. But you could also get asked questions about how cells like this are adapted for absorption. So you can talk about the increased surface area, so the infoldings, the microvilli. You can also talk about cells like this having high concentrations of things like mitochondria to produce ATP for active transport, and having high concentrations of ribosomes, rough endoplasmic reticulum, Golgi body for the formation of membrane-bound proteins like the dipeptidases and like the co-transporters and the protein pumps. And that's probably about it. That's all I can think of for now. So, well done for listening. That was a lot of information, a lot of new words, and a couple of brand new concepts that you need to get your head around. So what do I suggest that you do now? I suggest you first go over all of the new terminology. So check you understand what all these words mean. Check you can understand where these things are happening and why they are happening. So remember, it's not enough to just understand what's going on. It's important that you can pick it apart and understand why it's going on and how those processes could go wrong. Most exam questions on this topic are process-based. Describe the process of protein digestion or describe the process of protein absorption. But you can also get questions that are like experimental based. So, cells are exposed to more or less of something and then you get a change in absorption rate. Why? What could be happening? So think about this process from that perspective as well. But well done for now. Speak to you soon.